Es con gran humildad y cariño que compartimos con ustedes nuestra investigación. Mi nombre es Oscar Alejandro Pérez Escobar. Yo soy un investigador colombiano que hace 8 años trabaja en el Jardín Botánico de Kew, en el Reino Unido, como investigador principal. Mi especialidad son las orquídeas, pero pues en mis tiempos libres con un gran colectivo de colegas trabajamos en, investigación que pueda permitir informar políticas y como tener soluciones inmediatas a problemas que tiene la sociedad. Y para mí es un privilegio especial poder presentar ante ustedes hoy personas tan grandes como Wade Davis, Dennis McKenna, pero especialmente a nuestros hermanos mayores, las comunidades indígenas, que son en verdad los propietarios de esa planta sagrada, ¿no? I'm going to talk in mixed language, if that's okay. For us, and I say us because I represent that collective of researchers, it is a tremendous honor and privilege to be here with you, presenting in front of such personalities like Wade Davis, Dennis McKenna, but most importantly, our big brothers, the indigenous communities who are like the rightful owners of this plant. If you have any question at any point, just raise your hand. So, I'm also aware that we humans have a very short time span of attention, so I won't spend more than 30 minutes because we are all hungry as well. So that's a brief outline of our presentation. So I want to introduce our working team, our motivation, and then I have 3 pieces of research I want to share with you. Now, given, let's say, the significance and how special this meeting is, our collaborators and I chose to bring 2 items of unpublished research, 2 major breakthroughs, which I think can help, fomenting discussion, promoting discussion around the plant. Nuestros investigadores y yo, nuestro colectivo, dada la especialidad de esta reunión y su significancia, hemos decidido traer ante ustedes 3 proyectos de investigación, resultados, 2 de los cuales están sin publicar, que nosotros creemos pueden fomentar discusión alrededor de la planta sagrada de coca. And that's it. So this is our team. We are a diverse group of researchers from many different backgrounds. We take great pride on being mostly Latin American researchers, which can actually offer a fresh research on the coca plant and all its implications, right? We have, for example, the Director of Science, Professor Alessandro Antonelli, a very prominent scientist. We have also local taxonomy experts, like Fabio Andrés Avila y la profesora Rocío Cortés Ballén. And then we have different disciplines, but we are much more than this team. Over the 5 years when we started this research program, we have built a very strong collaborative network involving many different researchers from many different countries. Again, we have quite renowned scientists like Professor Susanne Renner, who is an expert on reproductive plant systems and other stuff. More recently, we have engaged in cooperations with Carmen, who is really good at disseminating and translating complex knowledge into common terms. And we are really keen on open to cooperations. So this presentation is also like a rally call to join forces and work together as one. Right, so there is obviously no doubt that indigenous people are like the rightful owners of the sacred coca plant. But then we are also aware that all the situation of illegality of the plant that was unjustifiably given to it has brought many consequences. So I have picked one example here, well, 2 examples. That's the first one. Guess I will use the magic stick. So what you see here on the left side is a map that the United Nations Drug and Crime Office produced about, let's say, the permanency of coca plant plantations, where yellow, reds, and pale tiles represent long-standing populations. And one interesting pattern that you see is that some of these populations sit in what we call biodiversity hotspots. So what you see here is South America, and right here in this area, we have the most diverse biodiversity hotspot in terms of plant diversity, which is called the Northern Andes region. And curious enough, some of these long-standing coca plantations sit on areas that are extremely diverse. So these plantations are basically resulting in what it is biodiversity loss. I guess you're translating. Solo para resumir, obviamente los pueblos indígenas son los propietarios de la planta sagrada de coca, pero todo lo que ha sido la denominación ilegal de la planta ha traído muchas consecuencias y yo he escogido 2 ejemplos. El primero de ellos es la pérdida de diversidad. Este mapa que está ahí muestra como plantaciones de coca que han estado por mucho tiempo ilegales, con el propósito de producir cocaína. Y esas plantaciones básicamente están en puntos de extrema biodiversidad a nivel mundial. Uno de ellos es la parte norte de los Andes, que es básicamente el punto más biodiverso del planeta en términos de plantas. Y pues, digamos, esas plantaciones están afectando esa pérdida de diversidad que es única, ¿no? And then one very good example as well is the loss of genetic diversity because of the establishment of new varieties, right? So, for example, you read the news and then you have something like narcos alteran la coca para hacerla gigante y persistente. So, there are some reports that there have been the appearance of like improved varieties that can produce more leaf, resulting, for example, in the increase of cocaine per hectare. And then one very good question is where all this diversity of genes, of the genetic codes are coming from to improve this diversity, right? We don't know. So that's a matter of research. That's something that we are very interested in solving. Otro, digamos, caso que pasa con el contexto legal de la coca es que han aparecido nuevas variedades que aparentemente pueden producir más follaje y por tanto incrementar lo que es la producción de cocaína. Y pues una muy buena pregunta es de dónde viene esa diversidad genética. Viene de plantas que son provenientes de comunidades indígenas, nadie sabe. Son variedades que se han robado, nadie sabe. Eso es, digamos, una pregunta de investigación que queremos resolver. Y esa es nuestra motivación. So basically put it like very simple. We want to produce baseline knowledge research that can help informing policies around the regulation and monitoring of coca plantations or coca leaves. We also want to assess the impact that the legality of coca is having on like native genetic diversity. When I say native genetic diversity, I don't only mean cultivated coca plants, but also sister species, right? And perhaps most importantly, we want to bridge society with science. We feel that one major problem we have is that there hasn't been enough communication between us scientists and politicians and the general population, right? So, by the way, in this presentation, there's going to be some kind of core science. We scientists like very much to talk in our nerdy terms. I wish I would be like Dave that can really, speak fluently and translate complex science into something very simple. That's a really talent you have, sir. So I will do a very strong effort to translate what we are doing, why it matters in very simple terms. So please bear with me, don't fall asleep. Básicamente nuestros objetivos son estos tres, que es como producir conocimiento base que pueda informar políticas de gobierno, que pueden ayudar a lo que es la desregulación de la hoja de coca, pero también como lo que es la parte de monitoreo de las plantaciones ilegales, que aún están dando. También es como intentar entender qué impacto ha tenido lo que es la denominación ilegal de la hoja de coca en los recursos genéticos nativos, ya sea diversidad silvestre, como Dawson explicó bien, hay especies emparentadas con la coca que no son cultivadas. Y también estamos hablando de los recursos genéticos de las plantas que son cultivadas por pueblos indígenas para su uso tradicional. Y lo último es también hacer un gran esfuerzo para conectar lo que es la ciencia que nosotros hacemos con la comunidad en general y los políticos. Porque creo que una parte del problema que hay es que nosotros los investigadores, con nuestros términos super nerd, no hacemos un esfuerzo para, decir, vea, nuestra investigación es esta y por esto importa. Right, so there is one first research item which is already published I'm going to share. I think as Dawson and Wade explained really well, the classification in Western science of coca has been based on leaf, how the leaf looks, the shape. And this is also to say that the research that we have conducted until this point, of course, stands on the shoulder of giants. The work that Dawson has made has set a really solid baseground knowledge for us to build upon and expand. The same as Timothy Plummer, of course. And then when you look at, let's say, the description of this species, one important thing that you can notice is that the difference between the different varieties or species is the leaf itself, the shape, right? Digamos que en la ciencia tradicional, la ciencia occidental, para nosotros clasificar y reconocer las diferentes hojas de coca, como Dawson explicó, que pues su trabajo ha sido muy importante para nosotros, el cual hemos construido nuestra investigación, es la forma de la hoja. Con esa forma, los investigadores occidentales han decidido o han, tomado la decisión de decir esta especie es tal, esta especie es tal, ¿no? And this is to remind everyone why taxonomy is so important, right? So when we talk about taxonomy, we are talking about a system that a Swedish person built by accident, says the history, somewhere in 1753, I think. That was a Swedish botanist, Carl Linné. And then what he did basically is, by accident, give a first and a last name to every species, like every species, right? So every species that is known to Western science has a first and a last name, what we call genus and species. So if you go back to the context of coca, as Dawson already explained, we have coca, naugranatense, all these taxonomy names. And these taxonomy names matter. And this is because huge amounts of knowledge are stored in these names, right? You can communicate to someone in China, even though you don't speak Chinese, by giving a scientific name. Then this person can really access information, right? And then, if you refer to the context of coca, these names have been widely used in reports. One example here on the left side is a report produced by the US Department of Justice about coca cultivation and cocaine processing. And as you can see here, we have all this bunch of scientific names. A more recent example here is a report by the United Nations Drug and Crime Office, again with scientific names, right? So this is extremely important, right? Para la ciencia occidental, un sistema de clasificación que fue inventado hace 250 años, cada especie conocida para la ciencia occidental tiene un nombre y un apellido. Y estos nombres importan porque una gran cantidad de conocimiento está almacenada en ellos. Y aquí hay dos ejemplos, dos reportes del Departamento de Justicia de Estados Unidos y uno hecho por la Oficina de Naciones Unidas de la Droga y Crimen. Right. And until this day, the shape of the leaf is very important to try to identify different varieties. This diagram that you see here is basically one built by police reports on the different varieties they have found. They have given their own names. You can see, for example, Boliviana Blanca, Pati Roja, etc., etc. And then one cool thing you see here is that the leaf, again, is one main feature. And then there's, of course, the size of the tree and so on and so forth. So the key message here is that leaf has been used for monitoring to understand which varieties are replanting, right? But then we thought about, as Dawson explained, we have now genes, we have now technology. We can compile many different lines of evidence to try to understand how stable these classification systems are. Then we thought about testing that. Can we really recognize the different varieties of coca by the leaf? So what we did was something very fun, which I will try to explain in very simple terms. And it's that we took herbarium specimens. I think that Dawson did really a proper job explaining what a herbarium specimen is. It's basically a plant that is dried and stored in a museum of dried plants, herbaria. We took a bunch of leaves, hundreds of leaves. And then what we did is that we tried to, by hand, crop each leaf and then take out the outline on this leaf in such fashion you see here. And then what we did is some fancy statistical analysis to try to convert these shapes into numeric variables, numbers. Then try to see how these different numbers relate to each other, something that we call principal component analysis. So if you translate these shapes into dots and samples where every dot is one leaf, it will look something like this, right? So let's say that in an ideal world, as a plant taxonomist, you would like to see something like this, right? You want to understand differences between species, and you would hope to find a landscape like this, where you compile all these shapes or colors or measures, if you like, and once you do the analysis I described, you will find that all these shapes will segregate into discrete clusters of points, like this. But that's an ideal world, right? And life is much, much more complicated than this, as we found. It's everything clear so far? Okay. So, because life is not easy and the world is not as we want it to be, we did the same analysis with hundreds. We did analysis with such hundreds of leaves. And remember, every dot you have here is one such leaf transformed into a number. And what we found is that you cannot really distinguish the varieties under the common current naming system. And perhaps if you can segregate one cluster here, is Gracilipes, but then it's really difficult to find out which variety is which by the leaf. So I want to highlight here the role of Rudy Diaz, who was my master's student some years ago. He's a brilliant guy. He came with this analysis, so kudos to him. Para resumir en español, lo que intentamos hacer fue como mirar si logramos separar las diferentes variedades de coca por su forma de la hoja. Entonces lo que hicimos fue tomar cada hoja, dibujarla y convertir la forma de la hoja en una variable numérica y luego intentar comprender cómo esos diferentes números se relacionan unos con otros, si se pueden comparar, si se vienen juntos o no. Y pues lo que encontramos es que no se puede. Y esto pues, investigación fue hecha con muchos especímenes de herbario, ¿no? And just to clarify, all the research that we are presenting here is mostly based on herbarium collections. They have a great importance. Mostly our herbarium at Kew, which have 7 million specimens. It's a bit hard to say, but all of these specimens come from, let's say, the time where the British Empire was in its full exploration mode. But without those specimens, we couldn't have done our research. So, as we say in Spanish, no hay mal que por bien no venga. But then something very interesting that we found is that when you consider certain aspect of the leaf shape, like for example, how it looks, if it's rounded on the base or on the top, you can find one difference between the cultivated varieties versus the others. This means that if you take a leaf of a coca and you find that it's mostly rounded at the base, it is likely one of the cultivated varieties. And then my colleague Natalia Sheomska, who at the time was my postdoctoral researcher and now is a lecturer at the University of Portsmouth, came with a brilliant idea that this could suggest a domestication syndrome. It's basically a trait that a plant acquires because we have selected for it. That is just one hypothesis that we have to test, of course, but it's a nice idea. And the hypothesis comes from the fact that if you have narrow leaves at the base, you can harvest them more easily, but it's just an idea. We have to conduct much research to test it and make sure that it's correct, right? It's just one hypothesis. Una diferencia que encontramos mirando las hojas entre las plantas cultivadas y sus parientes silvestres es que las plantas cultivadas tienen una base mucho más angosta y nosotros proponemos por primera vez una hipótesis que esto es un síndrome de domesticación, una característica que las plantas han adquirido por una facilidad que al humano le gustó, que en este caso sería como cultivar las hojas más fácilmente al arrancarlas, ¿no? O como decimos en Colombia, al raspar las ramas. Then we look into the DNA as well. So, as Dawson explained, we just took the genetic code of many, many hundreds of these leaves, and we tried to understand where we could segregate them as well by their genetic code. But what we found is that you cannot really list some of the varieties. So every dot here represents the genetic code, at least a little bit, of one leaf of one individual, right? And what you can see here is that there is a massive overlap between Gracilipes and one of the cultivated cocas, that is Cericoxylum coca. And if there is one cluster that you can segregate, is Novogranatense, which falls here. Then you have another wild relative over there, which is Cataractarum. Oh, and I forgot to mention something very, very important. If I go back here. Oh, well, you cannot really see because the text is cut. But then something that our research has pioneered is including what we call the type specimen, right? So that's a term that we use, but I will try to give a, let's say, simple example. Can I take one flower here? Say that, for example, one day Fabiola, who is not paying attention. Let's say that one day Fabiola is walking in her territory and finds, by chance, such flower, and that flower is unlike anything we have seen. So then, if you are taxonomist, we could see. I will translate in a minute. Ya traduzco. So assume for a second that Fabiola finds this flower which no one has ever seen, right? So you as a taxonomist, you could say, oh, this could be a new species, right? Something not known to Western science, but might be known to indigenous people. So if you want to give it a first and a last name, a scientific name, what you do is that you take that plant, you press it, and then you do a description, and you publish a new species, right? So this becomes the type specimen, which is basically the only link that you have with a scientific name and a piece of plant. Anything else than that is just an interpretation of a name. It's something that we think, oh, it's similar to the description of a new species. Un espécimen tipo es único en el mundo, es de gran importancia. Asumiendo que un día Fabiola en su territorio indígena encuentra una flor que nadie ha visto, que la ciencia occidental no conoce. Fabiola la puede describir como una nueva especie. Entonces esto, con base en este espécimen que Fabiola va a secar y lo va a poner en un herbario, es el espécimen tipo. Y es el único, digamos, asociación física que tenemos con un nombre científico y una planta. Y de este espécimen solo hay uno o muy pocas copias. And we decide to include type specimens given the ambiguity and also, let's say, the plasticity that leaf shapes have. So, in each of these plots, whenever you see something that says isotype or orotype, is one such specimen. And this is of great importance because for the first time, we can link not only morphology, but genes from such type specimen. So this is something very important. Right, now we are going to talk about phylogenes again, which doesn't explain. Just to briefly remind you, this can be seen as something extremely complex, but it is not. Think of a phylogenetic tree as a genealogical tree. It's the stuff that you build to understand who were your grandparents or your great-great-grandparents, and it helps you to really understand how things are related to each other. And for us, biologists, it's an essential tool to understand how speciation occurs. That is the means why a species form, right? So if you translate this genealogical tree, where the tips are basically the species that you see today, you can trace their ancestry and say, for example, Nougaratense and Coca had a shared common ancestor sometime in the past. So for us, biologists, this is an essential tool to understand, how biodiversity originates. Ahora voy a hablar un poquito de lo que son árboles filogenéticos. Puede ser visto como algo muy complejo, pero no lo es. Piensen en una filogenia como un árbol genealógico. Una cosa que uno construye para, por ejemplo, entender quién fue su abuelo o su tatarabuelo, etc. Y para nosotros los biólogos es una herramienta esencial para entender cómo la vida se originó en la Tierra. Así, simplemente visto. And this is when things get very, very interesting. Maybe this is obvious for any plant biologist or any biologist, but when you take a cell, when you look at a plant, right, like the flower that Fabiola was holding in a minute, you look at every cell here. This cell has 3 genetic compartments. These were basically the instructions to build how you look are included in interaction with the environment, of course. So a traditional plant cell has 3 genetic compartments. The nucleus has the nuclear genome, the chloroplast genome, and the mitochondrial genome, which is the powerhouse of a plant or an animal, or etc. And the crazy thing is the following one. When you try to understand how things relate to each other by looking at each of these genetic compartments, you often find that these genealogical histories are not congruent to each other. They are in fact incongruent. And this can help us understanding how life originated on Earth. So something that our research has pioneered as well is by looking at the history of coca through these different genomes or genetic compartments. Don't pay attention to this diagram too much because it's extremely complex. The message with this slide here is that when you look at the information, the genealogical history that the nuclear genome gives you, the nuclear DNA gives you, compared with a plastic DNA, which is another genetic compartment in a cell, they are very different. Can you see how the names are placed differently here? And this has led us to understand, or at least to suggest, that the way that coca has evolved is by exchanging genes with unrelated species. Let's say that the key message here is that if we want to talk about, regulation about the coca leaf, we have to think beyond the species that we are currently cultivating, because there is, let's say, native diversity in both as well. There can be gene flow between native species and cultivated cocas as well. Esto no lo voy a traducir en español porque es demasiado complejo. Pero digamos que cuando miramos una célula de una planta, cada célula tiene como 3 diferentes compartimentos genéticos. Y si uno intenta entender cómo la vida se originó, uno va a encontrar que la historia evolutiva de esos 3 compartimentos no es congruente. Y eso se ve porque a las plantas les gusta hibridizarse, cruzarse entre especies. Y cuando uno mira la historia evolutiva de la coca, desde uno de los, compartimentos, que es el nuclear, que es el más importante, uno ve que hay una historia evolutiva Pero si usted mira otro compartimento, que es el del cloroplasto, que hace que las plantas se vean verdes o rojas, etc., la historia evolutiva es muy diferente. Y esto nos ha permitido entender que en la evolución de las cocas cultivadas y sus parientes silvestres ha habido intercambio de genes. Las plantas son muy promiscuas, les gusta intercambiar genes entre otras plantas, etcétera, etcétera. Esto es normal, no es sorpresa para nosotros. Pero yo creo que, digamos, en lo que es legislación, para pensar en que, digamos, plantas se quieren desregular, hay que pensar más allá de la caja, think out of the box, y también pensar en discutir los parientes silvestres. Right, now to finish this part of this research, something else that we were quite interested in is trying to understand the timings of origin of the cultivated cocas, right? When did they appear on time? Did they appear when humans arrived to South America 15,000 years ago, or were there before us? So what we did is that we built this genealogical tree, but instead of, let's say, what we did is build this genealogical tree, and then we tried to understand how old this tree is in terms of time units, that is millions of years. And what we found is really interesting. When you try to see, for example, when Gracilipes diverged from Coca, and Nougaratense diverged from Cataractarum, which are like the wild relatives, you can actually see here, this branch is in time units, that the divergence of these species and populations goes back at least 200,000 years before humans arrived. Now, this doesn't mean that the coca, as we see it now, like producing copious amounts of different alkaloids, it was in the past. We don't really know when cultivated coca plants started to produce cocaine and many other tropane alkaloids, right? So that was also another major truth of our research. So this is to say as well that this understanding of coca, as we will see later, comes from a very small fraction of the genetic code of coca. So this is, in our opinion, inconclusive, right? We need more evidence to see where this story holds once we analyze larger parts of the genome or the genetic code of coca. Is everything clear so far? Está todo claro, diáfano, cristalino, como el agua del río Bogotá. Cool. By the way, this study was published last year. If you want to read the paper, it's super technical, but we have a debriefing, which is really written in lay terms. We will be very happy to share with you. Nuestra investigación fue publicada el año pasado, pero pues es muy técnica, pero también tenemos como que es un resumen que cualquier persona puede entender y estaremos muy felices de compartirlo con ustedes. Right, now comes to, let's say, my opinion, my humble opinion, the most exciting part of this talk, which is the stuff that we have not published yet, and that we are very pleased and humbled to share with you. So, as Dawson was explaining before, for us biologists to understand how life originated on Earth and how, flowers look red or yellow, we need to understand in great detail the genetic code of plants, what we call genomes. The term might sound very complex, but think of it as a book. So, a book can tell you a story, right? But if you take off pages, then the story will be complete. It would be a crime for me to take, parts of the pages of your book. That would be, a crime. It's the same with a genetic code. So, the more complete a genetic code is for us, the more understanding we have as to why, for example, certain plants of coca produce more cocaine or different alkaloids or not. So far, there has been only one such genome published with great contiguity, as in a book with all pages. That was published last year by some collaborators in China. We suspect that what they, let's say, sequenced or decodified is a plant of Erythroxylum nougaratense. But here I'm very pleased to announce that we have produced a second such genome from a plant cultivated in Leiden. It was a massive undertaking because of all the regulations that exist. So just to make the long story short, traditionally when we want to sequence a genome or decode the genetic code of one plant, we make collaborations, we bring material, we work together. But because coca is so regulated, we couldn't basically get material to Kew where we are based to produce a genetic code. So long story short, we had to travel to Leiden, which is where the plant grows, a botanic garden. We had to take our own machine to produce the sequences and do all the work there, which made everything more expensive, right? But such as life. Doing research in coca is actually very challenging, as you will hear from La Profe. This genome, in terms of completeness, let's say how many pages it has, is 95% complete. If you think of pages of a book, coca genome has 1.3, let's say, billions of pages. Ours is 95% that, 1.2, what we call bases, gigabases. If you try to see, for example, how many genes, that is pieces of genetic code that code for a function, or might not, we have that is 95% complete. Thanks. Well, this is years of research from.
What does it mean that they are missing? Why is it 100%? What is like
that is a very good question. So usually when you manage to have a complete book of a plant, it's because some parts of that genetic code are repeated across the genome, right? So to explain better, these lines, which you cannot really see well, but let's say that these numbers represent chromosomes. So chromosomes are structures where our DNA is packed, the nuclear genome, right? So sometimes some plants, for some crazy reason, and actually coca is one of them, their genetic code is heavily repeated. It has many bits that occur many times, the same bit across the chromosomes. And then because of that, because of the methods we have, to get one that is 100% complete is extremely complex. I mean, the human genome was just published completely just a few years ago. And is a human genome, right? Which is one of the most, say, sequenced organisms, I would like to say. It's because of the complex of the genetic code of a plant that we cannot easily have 100% in just one go. So usually what you do is that you publish a draft, say a draft of a book that lacks some pages, then you revisit that book again, and you produce more information and so on, and then you produce a second version, a more complete one. That is the way we produce a complete genetic code of a plant. Para traducir, el código genético, ¿no? Okay, cool. Because time constraints, right? And so to put things into context and to give a very important message, our current understanding on the evolutionary history of coca, which is the great work that Dawson has done and collaborators, come from these bits of genes or pieces of genetic code that you can see across chromosomes. So Dawson did a really good job trying to select good genes, good pieces of this genetic code to build a phylogenetic tree and understanding of how populations relate to each other. But if we put things into context, if we compare the 95% of the genetic code of coca versus what we have used for research so far, which is Dawson's genes, that might equate to 0.1% of the genetic code of coca, right? So in our humble opinion, for us to understand more about relationships, how coca has evolved, we need a lot more information. But that's how research is done, It's one iteration, then you produce more information, and then you move on. To move on our research, I'm also very happy to announce that we have sequenced 500 environment specimens. That is to better understand and hopefully help taxonomists like Dawson to produce more reliable classification systems. And in this new batch of data, which is enormous, right? We have also included more type specimens. So this plot that you see here is, let's say, a way to understand how these different varieties and their type specimens relate to each other based on their genetic code. I'm not going to go much into detail, but this is just to say that by including more type specimens, basically the only link that you have with a name and a plant, it will be essential to inform taxonomy, right? And understand how cocas have evolved in greater detail. And now to move to the last part of our research, we have also been quite interested in understanding the chemical diversity of cocas and their wild relatives. And often in the discourse, all that you hear is, coca has cocaine and so on. But as some of you know, coca produces many more alkaloids than just cocaine. I think if I understand correctly, up to 15 different tropane alkaloids, one of which cocaine is one of them. And what you see here is the research that we did based on 150 specimens that we analyzed. And there are some very interesting patterns here. This plot that you see here is basically how we characterize chemical compounds. We basically take an extraction of a leaf, we put it into a machine, and then we let that machine analyze all of these compounds across time. And so certain compounds, because they are bigger, they have, let's say, they take longer time to produce a signal, which we call a peak. And then the key message here is that when you compare, for example, the wild relatives, which is Cataractarum, Gracilipes, with the cultivated coccas and some outgroups, other species related to coca, you can see very interesting differences. For example, on the specimens we analyzed of Cataractarum and Gracilipes, this peak here, which we know is cocaine peak, is absent. But if you compare the cultivated plants, you have here a huge peak of cocaine. And what is even more interesting is that you have variation within individuals of cultivated coccas. So for example, here you have one specimen which comes from our economic botany collection in Kew. It's a plant that was traded to Java. It's a piece of leaf that has like 200 years or so. You can see that the peaks here change compared with our coccas that we analyzed. And when you analyze Novograndense, you can actually see that the peak of cocaine is smaller, which might suggest that this variety produces less cocaine, and then have similar peaks here. And last but not least, we have also tried to do research understanding why some coccas produce cocaine and why not. And there was some very important research from last year by Wang and collaborators, and then on 2021 by Chavez, who finally managed to understand how all the building blocks of tropane alkaloids go. As Dawson said, the way cocaine is synthesized is by building blocks, right? You start with a molecule, and then you modify it, and you add more stuff, and in the end, you finalize in cocaine, right? And the research that Wang, let's say the key message of Wang's research was that there are two genes involved, let's say two pieces of genetic code that are involved in the synthesis of cocaine. So these two genes basically produce an enzyme which is called cocaine synthase that add this ring here. An ester ring is a chemical organic compound that makes this compound here becomes cocaine. So what we did is that we compared with our genetic code, the genome, how this gene looks if we compare cultivated coccas with the wild relatives. And what we found is really exciting. So, sorry, it's my scientist nerd, kicking in. So this sequence that you see here is amino acid sequences. That's the gene itself from a cultivated coca. And what we did is that we compared the sequences of the reference genome, a cultivated coca, and then two relatives. And all these red arrows that you see here are basically modifications to that sequence. So when protein sequences have modifications, might suggest that the enzyme they might produce might not be functional, and therefore might result in, let's say, the absence of production of cocaine, or at least the production but in lesser amount of cocaine. So that is something that is really novel. I don't think that someone has ever done research on that and why the wild relatives do not produce cocaine. So we are really excited about it, and we hope that this can help informing policies as well. Good. And then just to finalize my presentation, as we said before, as a collective, we do really strive to try to translate the knowledge that we produce to the general population. And this is not an easy task, because sometimes when you engage with journalists, they have their own agenda and then they try to send their own message. But we have to start somewhere. So we make sure that all the research we do, we share with journalists and in different conferences. I would like to give a shout out here to Carmen, who is organizing a really cool event, Futuro Coca. I had the privilege to attend and share our research there, which has received massive attention. So if you have a chance to go to Carmen's festival one day, you should. We were also recently invited, actually as to last Friday, to participate in the discussion that United Nations and the World Health Organization is doing on the deregulation of coca leaf. We presented there our work. You have here some piece of news on our research. And if we translate it into numbers, that translates into a paper being one of the most, let's say, receiving online attention the most in the ones published in this journal, which is one of the top tier journals in the world in molecular biology and evolution. So these are our next steps. We have something very exciting working on. We have managed, in cooperation to archaeologists working in Chile, to access the genetic code of a piece of leaf that is 1,000 years old. So that would definitely help inform me, like trading routes and so on. And with the message that our research will produce, we hope that this will help people understanding that not only here in South America, but everywhere, that coca is a much, much more ancient crop and something completely like dissociated from cocaine than it is now. We want to include as well hundreds of samples that we have access to that were collected in zones of conflict and illegal plantations in our research to try to understand where these, let's say, varieties produced illegally or unsupervised come from, like where the genes are being taken from. And this is also to say that this is really simply too much work for us. While we are a big network, as I showed before, we don't have enough hands. We also need more resources to produce more data, hire more hands, to analyze the data and publish our results. So if you're interested in cooperating with us and working with us, please get in touch. We are more than happy to engage. And that's it. Viva la coca nuestra!